Hello, my name is Halla Hamdan and I am a master's student here at OSU CHS. I'm going to speak about the research titled Effects of Gram-Negative Bacterial Outer Membrane Permeabilization on Serratia marcescens Gene Expression. Using RNA-seq data, we demonstrated that Serratia marcescens displayed transcriptional changes when treated with the outer membrane permeabilizer compound 4880 as compared to untreated controls. Several genes were statistically up and down regulated. 161 genes were upregulated for the 30-minute time point, and 47 genes were upregulated for the 150-minute time point. There were also 176 downregulated genes for the 30-minute time point and 68 downregulated genes for the 150-minute time point. We noted a significant upregulation in SLY-B, Fo-P, and Fo-Q during the 30-minute time point, but not as much of an increase at the 150-minute time point. Fo-P and Fo-Q are part of a two-component regulatory system that responds to its environment and acts accordingly by modifying transcription of numerous genes, one of which is SLY-B. Our end goal is to perform qPCR on these three genes in addition to housekeeping gene candidates to observe how the cell is responding to compound 4880 treatment across various time points. We utilized numerous techniques to gather this data, starting with the PATRIC, or Pathosystems Resources Integration Center analysis tool. This tool allowed us to reanalyze the RNA-seq results as well as check the quality of this data. We identified significantly upregulated and downregulated genes through this analysis. Next, we looked at fragments per million reads, or FPKM, which is calculated for each read, to compare expression levels and to identify housekeeping genes. To perform such identification, genes within the Serratia marcescens genome had to show consistent expression, which also had an FPKM greater than 10 across the two time points, given that qPCR cannot find genes with an FPKM lower than that. Our next step was making primers for qPCR using the IDT PrimerQuest tool. And finally, we used the new PATRIC genome annotation to identify additional differentially expressed genes that weren't found in the RNA-seq analysis. Looking at the first table, this is showing great increase in transcription of the three genes SLY-B, Fo-P, and Fo-Q at mainly the 30-minute time point and a much lesser increase at the 150-minute time point. Tables 2 and 3 display the genes that weren't recognized in the earlier RNA-seq analysis along with their encoded proteins and their log 2-fold changes at both time points. Table 4 illustrates our primer sequences for the three genes of interest, Fo-P, Fo-Q, and SLY-B. Tables 5 and 6 show the quality control performed by PATRIC on each RNA-seq read. Each color represents a different level of mapping. Excellent or high-quality mapping indicates the read maps entirely. There are some reads that were unmapped and some that had only a partial matching to the gene. Table 7 shows some potential housekeeping gene candidates for the qPCR. The conclusions are that the quality of the RNA-seq reads were excellent. The new analysis results show that there were 133 upregulated genes from the 30-minute time point and 48 upregulated genes from the 150-minute time point. Furthermore, the analysis allowed the identification of several additional differentially expressed genes we hadn't seen in the earlier annotation and analysis. And finally, we identified some potential housekeeping gene candidates for qPCR. I'd like to thank Dr. Champlin and Dr. Katz for giving me the opportunity to assist with this research over the last few years. Thank you.